Good evening, bachon. Very good evening. How are you all? That's good. Today we will be going with uh, the last part of our lectures. That is RFLP, AFLP, RAPDs, and also the DNA fingerprinting. They were the most leftover parts of our work. So we will touch these two parts. Most likely we will end our class soon today, maybe by 9 or so. Uh, any questions you have? Any questions? Please feel free to ask any questions today. I will be happy to answer them to my best level. So today is the 8th day for workshop. Usually this workshop that I'm teaching you, it goes for one semester for 6 whole months. But we did this within 10 days. That's kind of very, uh, what do you say, um, uh, fast and furious, like that. Cool. So, so soon in 3 minutes we will start. Let all people join in. So today will be recording will be there, so you can have it for later also. Yesterday my computer was not working well, so couldn't make it. And students, stay at home. Don't go outside. Whatever the things most likely you have to go for, just try to isolate for this September at least, because this corona waves is again coming up. My real brother living in Chandigarh and his whole family got affected now. So I'm really worried about others also. So please, 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 it's my humble request to all of you. Try to stay at home. Don't try to go outside. Whatever things are necessary, just try to do that at home. Uh, Try to go for maximum learning at home only. Don't try to go outside, because India is at the second number now for this COVID thing, and most likely this will increase to go to number one. So keep that in mind. Good evening, bache. Good evening. So, shall we start? That's our first topic of today. So I will be dealing with the two lectures, one with the DNA based markers, RFLP, AFLP, RAPD, SSRs. Other the topic that I will be dealing is here with the DNA fingerprinting. That how could we use DNA fingerprinting in our field of plant biotechnology. So, so is the voice clear so far? Do I have to increase or decrease the voice volume, or how is it? Shall I increase my voice level? Okay, so I shall start now maybe. So, principles of plant biotechnology. Under that, our first topic for today is the DNA based markers, that is RFLP, AFLP, RAPDs, SSRs. Uh, so that's kind of your one of the last topics of your unit 2 now. So we have done with most of our unit 1, unit 2 in the last 10 days. So today is the 8th day of your workshop. In this workshop today, we will be dealing with molecular markers, basic principle of molecular markers detection, how you detect them. Based, then there are the divided into two types. One is based on DNA hybridization, one based on the amplification, PCR amplification. Uh, so so one are based on the PCR amplification, one are based on the DNA hybridization, and one is then there is a combination of both DNA hybridization and PCR amplification, that is RAPDs, that is a random amplified polymorphic DNA, RAPDs. Then further there are also sorry, the combination of both RA RFLP and RAPD is known as AFLP, that is amplified fragment length polymorphism. So in the RFLP, we use restriction enzymes to cut the DNA. To then the DNA hybridization or PCR amplification is done, whereas in the RAPDs we have amplification of our DNA is happening, and in the amplified fragment length polymorphism is a combination of both RFLP and RAPD. That is, the restriction enzyme is also used, and also amplification is also done with the PCR. So it's a combination of both. Then we will discuss something about molecular marker assisted selection also, MAS. So let's start. A molecular marker is a DNA sequence in the genome which can be located and identified as a result of DNA genetic alteration, mutations, and the base composition at a particular location of the genome may be different in different plants. So they are different in different plants. According to it, it works out. So these differences collectively called as polymorphism, that is having a different composition of your genetic alterations, and we can map them and identify them. So plant breeders work is to detect these genes and the molecular markers, so that this is not always possible. So alternative is that we find these markers and closely associated with the genes and inherit together. So some if we find some markers related to salt tolerance, drought tolerance, some herbicide resistance, and something like quantitative trait loci is there, QTLs, and within the radius of that, with the radius of 5 centimeter of related to that, if our markers are present, then that they are the best one to work for. So molecular markers are highly reliable and advantageous in a plant breeding programs. So molecular markers provide a true representations of genetic makeup at the DNA level. They are consistent and not affected by the environmental factors. Molecular markers can be detected much before the development of plants occurs, and large number of markers can be generated as per the needs. So these are the various advantages of these markers are. So let us see the basic principle of molecular marker detection. How do we identify them? So let us assume there are two plants of the same species. One is disease sensitive and other is disease resistance. So if there is a DNA marker that can identify these two alleles, first we have to the DNA has to be extracted. Then it has to be digested by the restriction enzymes and separated by the gel electrophoresis. The DNA fragment then can be directed by the separations by gel, as we said, gel electrophoresis. For instance, disease-resistant plants might be given the shorter DNA fragment, while the disease-sensitive plants might be giving the DNA fragment to be longer. So by this way, we can differentiate between the uh, uh, disease plant and disease resistance plant by checking their uh, restrict by cutting them with the restriction enzyme and checking their various level of length of their DNA. So they are of two types basically, that is DNA hybridization and based on the PCR amplification. So the DNA hybridization is the non-PCR based approach, and whereas the PCR amplification are the PCR based approaches. So this is the diagram of what we have seen, the basic principle behind it. Here you can see there is a disease resistance sensitive plant, and here is a disease resistance plant. And DNA extraction has been done, and DNA extraction has been done here, and then digestion by restriction enzymes. And then we check them on the gel electrophoresis. So what you see is shorter DNA fragment in the case of disease sensitive plant, and in the case of disease resistance plant we see longer DNA fragments. So by this way we can with these markers, molecular markers, we can screen with different types of genotype on the particular species of a plant. So further molecular markers based on DNA hybridization, that is the first type. So they are widely used in the market, but major limitation is that we require large quantity of DNA in this case, and we also use radioactivity assays in this, which is not good in this case. So restriction fragment length polymorphism (RFLPs) are used in this case. So this is the first one in the DNA hybridization, that is your RFLP. Full form is restriction fragment length polymorphism. So these names are quite important. Just remember these names: RFLP, AFLP, RAPDs. What are their different full forms? So RFLP is come under DNA hybridization. That is the first technology employed for detection of polymorphism based on the DNA sequence difference. So it's mainly based on the restriction enzyme. In this case, we use restriction enzyme as a result of a mutation and recombination of genomic DNA. An outline of RFLP analysis is given in the next figure we will see. And the procedure basically involves, first we do isolation of DNA, then we do the digestion of restriction enzyme, then comes separation by the electrophoresis, then finally hybridization by incubating with the clone and label probes that you're looking for. So first you isolate your genomic DNA, then you restrict it by digestion enzyme, then DNA pieces are cutted out, then separation by agar gel electrophoresis, then DNA pieces are transferred to membrane filter, then incubated with the clone and label probes, then RFLP bands are detected. So this is how all molecular markers based on hybridization works out. So this is an outline. So but on other side, DNA hybridization could be worked more specifically. Let's see one more example to check in the DNA fragments, DNA restriction enzymes. So in the figure 53.3, two DNA molecules from plant A and B are shown here. There is a plant A and plant B. So in plant A, mutation has occurred leading to the loss of the restriction site that can be digested by E. coli. So in the plant A, there is some resistance, some mutation happen. So whenever E. coli will be there, it will not cut it in the plant A, but in the plant B, it always cut. So the result is that when the DNA molecules are digested by the enzyme Hint3, there is no difference in the DNA fragment separate. So when because when Hint3 is cutting, it will cut in both part, but when the E. coli is there, E. coli will not cut in the first part, and we will not see the band in the further direction. So this result in the polymorphic pattern of separation. So then we run them on the electrophoresis, these two samples. So this is the monomorphic pattern with Hint3 enzyme. We don't see much differences. So plant A and plant B with the Hint3 they cut it at the same at the 200 something base pair, 250 maybe. Whereas in the case of plant A, it's cut at there, but sorry, with the E. coli in the plant A, it does not cut. It remains at 1000 base pair. Yeah, whereas in the B plant, it's not has any mutation, so it will cut at the two parts at 350 and 650 base pairs. So we see a polymorphic pattern here. So that's why it is called as restriction fragment length polymorphism. So we do restriction, there is a length, and then there is a polymorphism also. Further, based on the presence of restriction site, DNA fragments of different lengths can be generated using different restriction enzymes. So, like we have discussed in plant A and plant B, like how things happen. So, this is the in simplified manner the system of DNA hybridization, how it works. So, there is a plant source, then there is a DNA. Then from the DNA we do the restriction enzymes. Then with electrophoretic separation we separate them, then we do the southern hybridization. Then with the autoradiography, we check the bands, what and where things are working out. Then molecular based markers on PCR amplification. So PCR is a novel technique for the amplification of selected regions of DNA. The advantage with the PCR is that even a minute quantity of DNA can be amplified. So small quantity could be amplified, and the PCR based molecular markers requires only a small quantity of DNA to start with. So the result is PCR based markers may be divided into two parts. So now it's the second type, which is based on the PCR. So there are two types here. So one is a locus non-specific, that is random amplified polymorphic DNA, and amplified fragment length polymorphism. RAPD and AFLP, they both belongs to PCR. Then comes the locus specific markers. These are your simple sequence repeats, that is SSRs, single nucleotide polymorphisms, SSRs, and SNPs. So we will discuss each of them one by one under the case of PCR amplification. So random simplified polymorphism, that is your DNA RAPD markers. RAPD is a molecular marker based on the PCR amplification. So an outline of RAPD is depicted there in 53.4. So what you do is you isolate your DNA, then you denature your DNA, then you do annealing of DNA with template with primers. Then you attach the primers like we do in the PCR. Then complement strands are synthesized, and then further amplified products by gel electrophoresis and identified at the end. So by this way, from isolation of DNA with the help of PCR, you amplify your products and run on your gel electrophoresis to identify your specific marker. So that's the basic principle behind this. So there is a single short oligonucleotide primers can be arbitrarily selected, and the amplified products are separated on the electrophoresis and find it out later on. So what are the applications of RFPD markers in plants? It helps to distinguish between different varieties. It's based on a difference in the DNA sequence. RAPD have been used to identify nearly 15 commercial sunflower varieties. The new bean varieties also, like Phaseolus vulgaris, and further candidate of RAPD marker methods have been extracted for each variety of the thing. So 12 samples were analyzed using 60 different primers. So here you can see that for example, DNA from a plant allows amplification of sequence A, C, D. So there is amplification of a DNA sequence from the A, C, D, but not B. So this indicates that in plant A, primer sites for primers used not found in sequence B. So the primers that we have used for the PCR amplification, it's available in A, C, and D plant, but not for the B. Similarly, DNA alternation at one point of primer binding site for sequence A has prevented from being amplified when DNA from the plant 2 is used. So here you can see all others are amplified like this case, but in the second case it does not amplify. It's remain uh, at C and D, the plant 2. So this shows that something is wrong with the plant then. So RAPD markers are employed in the construction of genetic maps. Genetic maps of several plants could be model. For example, Arabidopsis, tobacco, which are the model plants in the case of plant studies. And RAPDs have been extensively used to create 15 linkage groups in coffee. Further, these markers are used in direct selection of desirable traits that you're looking for, and also help in the linkage of interest can be screened for at any stage in the breeding program. So RAPD and other molecular markers have great value in the selection for desirable trait in long-lived species, which takes long time for maturity and show phenotypic character. For example, avocado fruit quality can be assessed in seedling itself using RAPD molecular markers. So RAPD markers have been used to identify several disease plants in like RP94 genes is responsible for the resistance to stem rust in barley. So that gene is also identified by the RAPD as a marker. And also RAPD markers linked to heat smut resistance. There is a disease, and we have also characterized with the help of RAPD. And moreover, the controlling of heights in barley plant by specific gene has been located, that is the dwarfism gene by RAPD markers. So that's are the various applications of the RAPDs. Further, if we see that in tissue culture, uh, somatic hybrids involving protoplasm fusion requires through screening. However, screening of somatic hybrid is cumbersome. So therefore, RAPDs can be exploited in that case. RAPDs has been identified in duplication of germplasm. These duplicates are then discarded once no morphological differences were detected. Genetic diversity was carried out in set of 63 tetraploid sweet genotype, which comprises 24 Durian land races. Further, RAPDs have been used in a variety of identifications, purity in grain processing in food industry. For example, Duran variety of wheat, which is preparation of food products, and contamination of other products can be identified using these molecular markers. So there are plenty of importance uh, of your RAPDs there, but there are some demerits. Non-alleles are not easily detected. There's a reproducibility of results are quite constrained in this. Your RAPDs markers are dominant, and only half the genetic information are codominant information are codominant markers in this case. So markers based on PCR amplifications, based on nucleotide alteration in the gene, polymorphism of the amplified DNA sequence differ, which can be identified based on gel electrophoresis. So genomic DNA from two different plants often result in different amplification patterns. That is your RAPD. So, so then comes your next part, that is amplified fragment length polymorphism. Polymorphism. So AFLP is a novel technique which is involved for the combination of RFLP and RAPDs. So it involves both restriction enzymes and oligonucleotide adapters and their amplification by PCR. So we use both a restriction enzyme in this case, and we also used oligonucleotide adapters as a linkers, and their amplifications by PCR is also done. This technique combines the usefulness of both restriction digestion and PCR. So both are being used in this case. So DNA of the genome that you are looking for is extracted. It is subjected to go for section digestion by two enzymes, that is a rare cutter AMS MSCL and frequent cutter E. coli. The cut ends on both ends and then ligated to known sequence of oligonucleotide. They are both cut MSCL with the help of these two cutters, and they are ligated by oligonucleotides. So PCR then is done afterwards after pre-selection of the fragments of DNA which has single specific nucleotides. So by this approach, we pre-selected the amplification pool of fragments and can be reduced from the original mixture. In the second round of amplification by PCR, three nucleotide sequences are amplified. So this further reduces the pool of DNA fragments to manageable level. Then autoradiography is done to detect the DNA fragments. Use of radio labeled primers and fluorescent labeled fragments quick this AFLP work of ours. So here you can see we have a DNA molecule. Sorry, where's my presentation? There it is. So AFLP. So yeah, I was here. So we have our two DNA uh, strands, and this is our DNA molecule. So we add our rejection enzyme, where E. coli will cut on the left side 6 cutter, where MUCL is the right side 4 cutter. Uh, so on the E. coli on the left side, so it cuts from the 6, 1, 2, uh, sorry, 3, 4, 5, 6, 6 cutter, and this is 4 cutter. So by cutting in that, so we removed, uh, so cat A is there in this case, the 4 are remaining, and CTTG is there, and the rest is still there. Then we ligate them with the help of linkers. We join them together. So linkers are added on that side. Then PCR is done. Then after the PCR, uh, there is a corresponding DNA producing against our uh, DNA. So against T, A is produced, then against T, A is produced, then against G, C is produced. So AACC. So PCR is producing a new link to the your linkers. So by this, a new amplified region will be there with the colored region. So in this diagram, we can see AFLP, that is amplified fragment length polymorphism is happening. So the lowercase letters represent the sequence found within the amplified region. The colored region indicate the linkers. So the colored region, this one, the blue one, here are the linkers, and that's how they generated a new uh, PCR with a complementary things onto that. Then further, let's take it to the isolated plant DNA, to the restriction fragments. Uh, so we have our ligate adapters attached to it, and these adapters attached to the restriction fragment things in there, and then we add non-specific primers, perform the PCR, then we go for electrophoresis. So this is a systematic simple method of showing you AFLP. So AFLP is used in genetic mapping. Several economic importance are there, such as in the rice, barley, and wheat. We produce different combinations of restriction enzyme in this case, and are distributed through the genome. In barley, AFLP are located on the long and short arm of the all 7 chromosomes, and these AFLP markers exhibit strong relation between the number of markers per chromosome and length of chromosome. So similarly, in rice, there is a cross done between indica and japonica, revealed 50 AFLP markers which were located on the chromosome except the chromosome number 12. So these polymorphic loci that we have discovered could be common throughout the genome of this species and can be illustrated by AFLP technique. And we can identify these various markers with the help of your AFLP technique. Further, uh, existing barley map developed, a RFLP comprises of 157 RFLP loci and has been increased by adding 118 AFLP markers. More than that, the level of polymorphism detected by barley AFLP can range from 12.2 to 29% in different species. Then breeding for resistance is an important program in cereal research, and they have find map of the resistance gene location delimited 30 kilo base pair. So these are the various merits of AFLP. That is, AFLP is used to screen large number of polymorphism. It is possible to saturate specific region of the genome of map-based cloning. Further, AFLP is used to screen pools of plasmid DNA from several clones, enabling rapid isolation of genes tightly linked to markers. AFLP has been recently applied to the analysis of quantitative trait in the barley and rice also. Further, AFLPs rapidly become preferred molecular technique for several different investigation, particularly many areas of research. So it has many merits. So some demerits are also there, such as they are not reliable to convert these AFLP into a CR. We will come up about the CRs. What are the CRs? None allele cannot be detected. Proprietary technique, intellectual property rights are being hampered in this case, and requires a high amount of DNA than required in the RAPDs. A relatively quite expensive technique owing to the requirement of silver staining and radio and non-radio labeling things. So quite expensive as compared to others techniques. So on that point, we are done with the RAPDs, RFLPs, AFLPs. So we are done with the three different markers. Now comes the marker based on the PCR amplifications on the second type of PCRs. So we are done with the RAPDs and RFLPs in that case. So now comes the STS, that is sequence stack sites. So they represent uniquely simple copy segments of genomes whose DNA sequences are known and which can be amplified by using PCRs. So STS markers are based on the polymorphism of the simple nucleotide repeats, that is GA, GT, CAA. So they are repeating in the case of plants and have been recently developed in plants also STSs. Their loci contain simple sequence length polymorphism SSLPs that are quite high valuable as a molecular markers. They have very quite importance. And many STS loci have been analyzed and studied in number of plant species. Many things have been studied so far related to that. Further, um, in the microsatellites point of view, microsatellites are the tandemly repeated multi-copies of mono, di, tri, tetranucleotide motifs. So you have your motifs of DNA. So they are the repetitions of the same, repeated copies of of all. In some instances, flanking a sequence of the repeat sequences may be unique. Primers can be designed for such flanking sequences to detect the sequence tagged microsatellites. This can be done by PCR. Okay, so sequence characterized amplified region SCARs. So SCARs are the modified forms of STS markers. They are developed by PCR markers that are made for the ends of RAPD fragments. So STS converted RAPD markers are sometimes referred to as SCARs. SCARs are useful for the rapid development of the STS markers actually. So then comes the fourth one, SCARs, that is sequence characterized amplified regions. So they are the regions that are modified forms of STS markers, and they are developed by PCR markers that are made up of end of the RAPD fragments. So STS converted RAPD markers are sometimes referred to as SCARs and are useful for the rapid development of STS markers. So here you have amplified with the primer RAPD band, and then it is amplify scar with the primer further. First you have RAPD band, then you amplify it scar with the primer further. So this is how you do your SCRs reactions. Now comes the last part, that is your molecular marker assisted selection, MAS. So what is MAS is basically done to check breeding programs. It's done to predominantly identify the phenotypes. It is now accepted fact that phenotypes do not necessarily represent the genotype. Many times the environmental make the mark of the genotype. So molecular marker assisted selection is based on the identification of DNA that you are choosing. So these traits include resistance to pathogens, insects, tolerance to abiotic stresses, and various other qualitative and quantitative traits. So the advantage with the molecular marker is that plant breeders can select a suitable marker for the desired traits which can be detected in advance. So following are the major requirements that you need in the plant breeding. That is, they have to be closely linked with the desired trait. The method should be efficient, reproducible, and easy to carry out. And analysis should be economical. Further, molecular breeding. With rapid progress in molecular biology and genetic engineering, there is now possibility of improving the crop plants with respect to yield and quality. So the term molecular breeding is frequently used to represent the breeding methods that are coupled with the genetic engineering and techniques. Improved agriculture to meet the food demands of the world is high priority area. For several years, the conventional plant breeding programs have been developed to grain yield and cereal production. So they are dwarf and semi-dwarf varieties have been produced of rice and wheat respectively during the Green Revolution, and this will be the all result of the molecular breeding, all these development that is happening. So there are some different terms are coming up for us. Molecular assisted selection was there, plant breeding is there, then the linkage analysis is there for us. So linkage analysis includes basically deals with the studies to correlate the link between the molecular markers and desired trait, and this is an important aspect of molecular breeding programs. Linkage analysis has to be carried out among the population of several generations to establish the appropriate linkage. So in the earlier years, linkage analysis was carried out by use of isoenzymes and associated polymorphism. So molecular markers are now being used for the same. Then QTLs, they are the quantitative trait loci. Just remember all these terms that are coming at the end. They are quite high importance because you will use many questions are asked related to these terms also. So they are this whole topic is quite hot actually. You have to remember the terms, what are their meanings are, what are the functions are, what are the various merits are, what are its various demerits are. So everything is highly demanding and quite important from today's lecture actually. So why we are studying all these things, what is the importance of all these things. So keep that in mind everything. So QTLs, these are the many characteristics controlled by several genes in a complex manner uh, and are, for example, growth habit, yield, adaptability to environment, disease resistance, all these things have been taken care by QTLs. So they are referred to as quantitative traits. The location on the chromosomes of the on the genes of these are regarded as quantitative trait loci. If you find these locations in the chromosomes, then they are known as quantitative trait loci. So they are in the full form QTLs. So major problem with the plant breeder phase is to how to improve the complex characters controlled by many genes. Now it is easy job with the help of genetic engineering. Therefore, it is very difficult and time consuming process. For instance, development of golden rice involving the insertion of just 3 genes took about 7 years. So, so that's it for this lecture. Let's take a 10 minutes break, then we finish with the next part of our studies. Yeah? Or shall I continue and finish it today? Just let me know how shall I go, students. So there is a small topic left for us now from our last part of it, that is uh, DNA fingerprinting. So shall I continue or do you want some break now? What do you suggest? Students. Okay, if you I just take a 5 minutes break and then I come back and we finish the things then. I hope we are done today. So class, I'm back. Uh, uh, let's start the next topic. So DNA fingerprinting. Uh, this is done to check also in the crime scenes, and also done as we see in the DNA fingerprinting in the case of plant diseases to check between disease and non-disease plants. So all these things are being done in this case also. So DNA fingerprinting is a way of telling individuals of the same species apart. Uh, a DNA sequence are variable and can therefore be used to identify characteristics of different gene sequences. And it has advantages over the source of evidence, and it is highly accurate and can be gathered from the trace or crime scene evidences. So how do we take DNA fingerprints? So here comes your the role of RFLP here. So one way you can do it section fragment length polymorphism, that is RFLP. Then there are molecules that cut into DNA pieces. So each enzyme is cut at very specific DNA sequence. So while all human beings share roughly the same DNA sequence, so they are in fact with a small number of differences, and these differences can be detected by the restriction enzymes. So this is how your RFLP restricted fragment length polymorphism is working. So you have your BAM H1 restriction enzyme which cut at GATCC in between G and other things. So here you can see that your restriction enzyme came and then it started to cut like this. Then you have your sticky ends. You don't have any blunt ends anymore, but you have your sticky ends. So these individuals uh, will have, so let's say this is individual one, and our restriction enzymes cut at the four different sides of it. And after cutting, it gives you five different fragments, that is A, B, C, D, and E. Whereas the individual two, it's cut at only three fragments, giving you mainly four fragments, that is A, B, C, and D. So there's individual 1 and individual 2, uh, which has different sides of section fragments. So summary, all in all, in this DNA fragment that we have studied is that essentially once the DNA has been cut by the enzyme, we have the DNA fragments by various sizes. So each individual band pattern should be different because of friction enzymes cutting at the different points for the different person, and the fragment of different sizes will travel along the distances along a gel where the electric current is passed through it. So then you can see the results of a different individuals. Let's say they are like now uh, different, one, two, three, four, five, six individuals are there, and six individuals are producing six different band patterns, and which we can identify in this case of gel electrophoresis having a different patterns. So this person size comes towards this, and this much, this is this much, they are quite close, they are quite separated, and this is quite big, these are two bigs come together. So these are all happening because of the restriction enzyme cutting at the different region of the person at different length. So this is something to deal with the forensic part. Just I want to add that maybe it could be used because mostly DNA fingerprinting is being used in the forensic science in the case of law and check the fingerprints. So just want to share that part to connecting to that part to the fingerprinting part. So DNA fingerprints show unique patterns from one person to the next, and that's how you can disputes use in paternity disputes and forensic evidence. Specimens collection should be licked, envelope, dirty laundry, cigarette butt, saliva, all are the important part of your specimen collection. And further, you have to take special care while wearing a gloves, disposable instruments, avoid talking and sneezing, avoid touching sample with your skin, air dry the evidence before packaging, mold does not grow. So enemies of evidence, sunlight, high temperatures, bacteria, moisture, all these includes your uh, preparing of DNA fingerprints. Then ideal sample is 1 ml of fresh and whole blood treated with ETA is ideal sample to test your samples. So again, here AFLP is working out to cut it into different organisms. So individual 1 is giving 3 and individual 2 it's giving D and C, two different parts. So that's how you see RFLP in the gel electrophoresis. If they're cutting 3 parts, you see A, B, C, and in this case C and D only 2 parts. So that's how the difference of individual 1 and individual 2 after the restriction enzyme element and gel electrophoresis, you can see the different results of your pattern. So this is showing you the gel electrophoresis with the agarose gel and probe with the hybridized ions. So this is the hybridized one, and this is the agarose gel at the when the membrane has been transferred. So you can see some patterns being seen. Like here we see different pattern, and here we see almost same, but there's one band here also coming up, and these bands are coming up like this. Light bands are there. So everyone has its own meaning. So first setup to do so is that you have to do southern blotting because we are working with the DNA part. So southern blotting will be done in this case. So that's how the blotting techniques that we did yesterday and restriction fragment lecture we did yesterday. So they are all being used in today's lecture. So their applications are being applied in today's lecture in every case of your studies. So first step in preparing a southern blot is to cut your genomic DNA and run on agarose gel. So you cut your DNA with a section enzyme and you put them on the separate them on the gel, and then you transfer to your nitrocellulose membrane. Then after transferring to the nitrocellulose membrane, uh, transferred radio label probes will be incubated. Then we can check that okay our bounded DNA exposed on the films were these parts. So last part is expose them radioactively label membrane to large sheet of film, and we visualize them in the probe to the DNA part like this. Then there is a VNTRs, variable number tandem repeats. So they are the sequence that are repeated multiple times, and the number of repeats varies from person to person. So there are 5 repeats like GATC, and in some person there are 2 repeats GATC. so they're being repeated in a particular things. So they occurs mostly in the introns, not in the exons. In the intron sites they are happening, and we can see them amplified by PCR run on agarose gel to produce uniquely DNA fingerprints. And we can see these different 5 repeats and 2 repeats of our previous sample, and we run them on the gel, and we can see them on the gel by this size pattern. Because on the top is the large DNA proteins are large DNAs are present, and at the bottom are the small DNAs are present. So that's how I showed you also the virtual lab of gel electrophoresis. So that's how the importance of these gel electrophoresis, PCRs, DNA isolations are so important, so that we can apply their applications into daily routine of our work. Then after checking it, you can do PCR to amplify the segment of DNA. So all what we need is need DNA primers, DNA polymerase, and nucleotides for to run our PCRs, and we can check our yeah results of our things like this. So reactions are placed in a machine called thermocycler, which goes at different temperatures as we remember, 95, 50, then 72 degrees Celsius. So first is 94 to cut into single, then annealing is happening at 50 degrees Celsius for primers, then at 72 degrees Celsius for a minute or two polymerase along the new DNA nucleotides are going on. The thermocycler repeats the denaturing, annealing, and elongation temperature approximately 30 times. So PCR amplification is logarithmic, meaning the number of copies of the target is doubled every cycle. So they got increased with each cycle. So, and what are the applications of it? It could help to diagnose various diseases, uh, and paternity testing it could help in it, and further forensic sciences as we discussed to check various loci. Uh, so we can check with the help of DNA fingerprinting many things. So there is one animation present. No, there is nothing. Sorry. Sorry, it's not working out. So also helps in the genealogy of animations. So like from where your DNA marker, your RFLPs were crossed, like once they were in Africa, from Africa, these RFLP markers cross to part of our Middle East. From Middle East, they go to the Russia part and also to the China and India, and then they transfer to the Australia. So while transferring these people, so there would be a chance of mutations were happening. Many changes happen according to the geographical region, according to the temperature and the region of food is there available to them. So people got to have different mutations in their body, and we can check with the help of RFLPs. So these DNA fingerprinting are a quite valuable technique to understand how, like from northern Canada to South America, so how things got changed uh, after the migration out of Africa in 60,000 years ago. Will this will work? Tried actually. Oh, sorry. They are not working. So don't try these links. So these are the genealogies that you can discover. So I think that's it for today. So I will send these lectures to you. So students, Photoshop does not work. So any questions? So I want to keep it small today because otherwise we don't have lectures for tomorrow. So I want to keep it this in 1 hour pace for our lectures, and we finish it. We do some more important part of it related to that. So I was thinking to teach you tomorrow maybe with the bioinformatics part and little bit about application and mass molecular assisted selection. So I will be dealing with that part tomorrow also. So these two things will be touched tomorrow. So in meantime, please also subscribe to the channel. That will really help me out to increase my yeah, my motivation to create videos like this and how we can improve them more. So please be with us and try to improve these workshops, and yeah, just like, subscribe and share. Okay, I hope today's lecture were quite confined and straight to the point, not so difficulty level. It was to the point and easy. Okay, if you have no questions, uh, so we stop it today, and then we see us tomorrow then, because all in all we are um, we are done. We are almost finished today. So so this part is done. Left mass and its application in crop environment. We will do that also, and some of the transgenic plants and their applications. We have already discussed many transgenic plants, so I will see if I could prepare some lectures for that. So just small two topics are left, and we also done the practical part one day, and these are all things are done. So we are almost finished for today. And how's your review writing going on? Are you writing reviews? How many you have to write reviews? So please write me back. Send me your reviews. We will try to find some journal to publish them. I will look for it if you can have. Students, please reply yes, no, something, any comments. Or is there any glitches happening? So you can't reply maybe. Okay. So in last 2 days we do some more parts related to bioinformatics. I will share some MBBT techniques with you and some bioinformatics techniques. So because we are here to learn, and I try to compensate with that with more stuff for tomorrow. Yeah, we arranged. I understand. We arranged for 2 hours class, but as I see the syllabus is finished, that was my main task. So I didn't thought that we will finish so fast in that manner. So maybe in the next time when we are trying to arrange workshops, we say 1 hour is enough for one day, because in meantime we are covering most of the topics of your essential plant biotechnology. Okay, you started. That's good. It will take some time. No worries, take your time, take your time. And try to publish uh, your review also. Yeah, whatever you have done, studied, try to publish that part also. That will be the best part for you. Okay, students, so my last thought for today is please tell to your family and your friends, family, friends, everyone to stay at home and don't go outside for at least September month. Uh, Many of my friends, my relatives, they are coming across with the corona positive, and it's the second phase is coming up. So please, 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 my humble request to all of you, stay safe as much as possible and try to isolate yourself from now onwards and don't go outside. Even though you're going, change your clothes after coming back. Wash your hands, take a bath or something. So this is about your life, and I really want that everyone, no one should get into that. Everyone lives matters. So please take care of that. We are the second number of the COVID. I started the same thing at the start of our lecture. I will also end the same thing. So please take care of that also. So okay, thank you very much. Then we see us tomorrow then. Good night. Take care.